Welcome to the Performance Around the Clock podcast. I am Professor Satchin Panda, a scientist in the field of circadian rhythm. In this podcast, I will talk to experts in many different fields about science, health, and human performance. You'll also find out why circadian rhythms is my favorite topic. I hope you enjoy the podcast and thank you for listening. Welcome to another episode of Performance Around the Clock. And I'm still in Lisbon in this Cell Symposium on exercise metabolism, and I'm super excited to discuss age-related muscle function. This is an area where if you look at any large scientific study, then you'll find the name of Bret Goodpaster. In fact, everything that we hear from all these health gurus online, they must have read at least one, if not many, of Bret's publications. I'm super excited. Welcome, Bret.
Thanks, Satchin. It's good to be here. Thanks.
Yeah. So, one thing is, why did we get into science? So I always ask people how...
Yeah. Why did I get into science? I guess it was just from an early, from high school, college, curiosity about the human body. Thought I might go into medicine. I was a biology major. Pretty clearly was interested in cardiometabolic health. My father had premature heart disease. So growing up, it was that sensitivity around heart disease. I was an exerciser as a kid. And then that became just sort of part of my lifestyle. And I had sort of two motivations, right? It was my own personal interest about my own exercise and performance, but also, how can exercise, prevent disease and improve health? Yeah.
So what did you do your PhD and postdoc? Yeah.
So I did a PhD at Ball State University at the Human Performance Lab.
Yeah.
Studied with David Costel, who was really in the 1970s made a name for himself and his lab by studying, some of the elite marathon runners, did a lot of sports nutrition studies. And so, that was the early 70s was at the time in the US was when marathoning was becoming really popular.
Yeah.
So he kind of popularized sports nutrition and exercise physiology that way. And so it was just, a really classic, lab to go study in.
Yeah.
And then after that, I went and did a postdoc at the University of Pittsburgh with David Kelly, who was really at the time in the, mid 90s, a rising star in the area of insulin resistance and diabetes and obesity. And, I sort of cut my teeth on, working with him and this whole concept of metabolic flexibility in diabetes and obesity. And then my interest with exercise really, was a good synergy with that.
Yeah.
Right. So, yeah.
So you used this term, metabolic flexibility, and recently you wrote a beautiful article in Cell Metabolism. Can you explain what is metabolic flexibility for general public?
Yeah, sure.
Yeah.
I mean, the basic concept of metabolic flexibility is that it's, your body wants to adapt and use energy according to, the prevailing conditions. Like if you're fasted, the body should be, and a healthy body should be very well set up to use and oxidize fatty acids for energy, right? You eat a meal, you get an insulin response that stimulates glucose uptake and storage of glucose, utilization of glucose. So that transition from fasting to feeding, being able to use the appropriate energy source is part of metabolic flexibility. And I would also argue that going from rest to exercise and being able to adapt and use the appropriate energy for exercise is also part of metabolic flexibility.
How do you measure that in lab condition and also in real life?
Yeah, you can measure it in the lab by measuring, grams of carbohydrate, grams of fat that are being oxidized during fasting, during, after a meal. During exercise. So it's pretty routine, non-invasively, to measure indirect calorimetry. in the real world, it's a little bit more difficult, right? But I think, with the development and the more common use, even among endurance athletes, of glucose monitoring, and maybe even, ketone monitoring, I think we could start getting into this idea of, being able to measure metabolic flexibility in the real world in, real time, right?
So that means a person who is, say, fasting, say, 14, 16, 18 hours,
Yeah.
that person's ketones should slowly rise up if there is metabolic flexibility. Is that
Well, that's a great,
partly this?
it's a great question. I think one of the unanswered questions, we've got some data on this, but it's not really, I wouldn't say it's that solid yet, but it looks like there are some people who can oxidize fat very well and don't produce ketones.
Really?
And other people do produce ketones. So it's not a direct correlation between enhanced fat oxidation
No, that's a
and ketones, right?
That's a good point, because a lot of people out there are crazy about ketone. They're measuring ketone and they're thinking ketones are the elixir of health.
Yeah.
But it's good to know that there are some people who can oxidize fat, which is the good thing about metabolic flexibility. They can, but they may not produce or increase ketone level in their blood to a point that is detectable
Exactly.
by a consumer grade keto meter. Let's put it that way.
That's right.
Yeah.
Exactly.
So, so in real life, then it's very difficult to measure metabolic flexibility.
Yeah, I think it is.
Yeah. But then going back to, you work on muscle.
Yeah.
And also fat.
Yeah.
So can you tell me a little bit about, people always think about building muscle mass.
Yeah.
And people can look at their maybe calf muscle and then say, oh, this guy has really good muscle mass. But at the same time, there's muscle and fat there.
Right.
So can you tell?
Well, I think that gets complicated too, because, the basic concept that, more muscle is better, I don't think is always true, right? Because we know that obese people with and without diabetes have more muscle mass, right? Along with their greater, BMI and greater body fat, they also have more muscle mass, right? So I think it's more important to consider the quality of the muscle, right? How strong is the muscle, how fatigue resistant it might be, rather than just focusing on the muscle mass. And we've done these studies on aging, clearly showing that muscle mass is really all that not that important with regard to overall function. It's really more about the quality of muscle.
So that brings up this health ABC study, Health ABC.
Health ABC, yeah.
Yeah, so can you tell a little bit about how many people were in that study and what was the age range? What was
So Health ABC study, Health Aging and Body Composition study, was an NIH-funded study that I was involved with from the beginning. And we followed this group of 70 to 80-year-old men and women over what turned out to be 16 years. And we started out with 3,000 subjects. And we looked at changes in body composition, changes in muscle mass, changes in function over time. And, one of the key findings in that study, among many, were that over time, people lost muscle mass, which we hypothesized. They lost strength, which we expected as well. But as it turned out, the loss of strength was 3 times greater than the loss of muscle mass. So
that means there are some people who had muscle, but they did not have strength.
Exactly. And that's really an important observation, because even, among these 3,000 subjects.
So I must say, 3,000 is a really large number for a human study.
Yeah.
And, we once in a while we see studies that are on 20 people, 10 people, and then we make big deal out of it.
Exactly.
But this is something, these are the kind of studies that give us biological insight and also knowledge about policy.
Yeah.
It
really allows you to start looking at the variability, right?
Variability too. Yeah.
And maybe not even so much on an individual level, but as a group of individuals, right? I mean, so Health ABC was important because it was an oversampling of African Americans, and for example. So we were able to study the effect of race on some of these age-related changes.
So let's begin with male and female in health ABC study.
Yeah, so it was about half and half men, half women. You know, the aging response was essentially the same in men and women.
So men and women.
They both lost muscle mass, they both lost strength. But what was important was that some of the people actually maintained muscle mass. And some maintained strength a little better than others. And I think, this is really important because we always talk about healthy aging, right?
Yeah.
And clearly there's different trajectories of aging, right? Some people age better than others, right?
So how do you define health from a muscle point of view? What is healthy aging?
I would say it's the muscle quality, right?
Muscle quality,
the strength of the muscle, yeah.
And how are you measuring muscle strength in health ABC studies?
So we measured leg strength and grip strength.
Yeah.
Leg strength was really the key because it more strongly associated with mobility limitations, long-term changes. So I think the leg strength, your ability to walk around was really important for these older people. Then the other important observation we had along the way was that, when you looked at muscle, it wasn't just pure muscle tissue. We noted and measured this fatty infiltration in muscle. It's like if you see a fatty steak at the supermarket, right? The marbling of fat. And that's exactly what you see in people too. Marbling of their muscle.
So this was, of course, you're not taking a slice of that muscle, so you're
imaging slices of the muscle, right? With CAT scans,
CAT scans. And here is this, I think many exercise physiologists have described this athlete paradox.
Yeah.
Can you describe what is that?
Yeah, so the athlete's paradox, to be clear, it's not really the marbling of adipose tissue or fat in the muscle like you see on imaging, but the athlete's paradox has more to do with I would say the microscopic fat inside the muscle cells, right? We know that fat stores fat in the form of triglycerides, and in the early 90s, animal models and even human studies had made these observations that the more fat in the muscle cells
inside the cell
was related to type 2 diabetes, insulin resistance, which is a hallmark characteristic of type 2 diabetes. And that was becoming the dogma in the field, right? That fat accumulation in the muscle cells was a bad thing. So the athlete's paradox was showing that when we looked at the muscle biopsies, the muscle cells in these athletes, they also had a lot of fat in the muscle cells. But they were low risk for diabetes because their exercise. They were insulin sensitive, not insulin resistant. So I think that led us and many others in the field to really start diving into what are the good fats and the bad fats in the muscle that might explain this insulin resistance.
So do we know now what are the good fats and bad fats in muscle?
Not exactly. I think.
What is the current hypothesis?
Yeah, the major hypotheses are that these other fat metabolites like disylglycerol, ceramides, and other sphingolipids might be the bad culprits here of insulin resistance. I think the jury's still out. Some studies, including ours, have shown associations with some of these lipids with insulin resistance and not others. Looks like it depends on where in the cell they are.
Really?
Yeah, like if they're in the membrane versus the cytoplasm. So the story is a lot more complicated.
Yeah. But then athletes, they store that fat or they're using that fat for the next run?
They're absolutely using the fat for their next run, yeah. They're storing it in the form of triglyceride, and we've done studies showing that when people exercise, and you don't have to be an athlete, even obese sedentary people, when they exercise, they're also using this muscle fat store for energy.
Yeah, so that means the muscle is designed to grab some fat and keep it for fight or flight kind of response.
Yeah.
And when you don't use it, that fat slowly becomes bad and gives you.
That's the hypothesis.
That's the hypothesis.
Exactly.
And then during that process, that fat may get converted to different bad fat stiff.
That's right. That's the hypothesis.
That's the hypothesis.
Yeah.
And then in this health ABC study,
Yeah.
as we age, we lose muscle mass, and a lot of people, what is the ideal or unhealthy rate of losing muscle mass or muscle strength?
Yeah.
What can people actually do to measure whether the muscle strength is appropriate
Yeah.
or worse? Yeah.
Yeah, I mean, I think it's hard for the everyday person to measure their strength. I would just say that they can sort of feel their strength if they're having difficult, walking. They can get they can get tested.
But I think in the paper most studies use this 400-meter walking test.
Yeah, the 400-meter walk test is sort of it's not just muscle strength and function, but it's cardiovascular system. It's sort of this integrative measure of whole body function. I would say what more closely measures muscle strength probably than any other thing is simply getting up out of a chair.
Getting up out of the chair.
Rising up out of a chair.
Yeah.
That's a pretty good indicator of your leg strength.
Yeah.
Or climbing upstairs.
Yeah. 10 flights, 20 flights of stairs.
Yeah.
But the inclusion criteria was they should have at least some mobility, right? For
Yes.
ABC study.
They had to be able to walk without a device to get in.
Yeah.
Of course, they started out between the ages 70 and 80.
Yeah.
And over 10 years, many of them became like immobile or mobility limited, let's say, right?
Yeah.
So, yeah.
And then during that study, was there any difference between those between 70 to 80 and the old adults? So, for example, since you followed them for 16 years.
Yeah. It's interesting. I mean, I think it really goes back to what we were talking about earlier, Satchin, which is this heterogeneity of aging, right?
Yeah.
Some of these people aged very well, and when they hit 90, they looked very healthy.
Yeah.
And others, had a sort of a steeper decline, right, in terms of their function. We studied some of the oldest when they were 90 years old, when they came back 16 years into the study. And, of course, you have to be healthy to come back into the study, right?
Yeah.
You have to be living.
Yeah.
So there's a survival effect here.
So how many people survived for the last visit? Not survived. How many people came back?
Yeah, it's a good question. We did a full 10-year visit, and I think of the 10-year visit there were
Couple of thousand?
about 2/3, almost 2,000 came back after 10 years.
Yeah.
So that's
good.
Pretty good.
Yeah, even 2,000 people.
Because you figure, the youngest person was 80.
Yeah.
And the oldest person was 90,
90.
right?
Yeah. So now you can differentiate between these 80 to 85 and 85 to 90.
That's right.
For example. And did you collect any data on that disease condition?
We did. Yeah, we did.
They're all over the place.
All over the place. I mean, lots of age-related diseases, more cancers, more cardiovascular disease, of course, right? Arthritis.
Yeah.
We did some arthritis studies. Yeah.
And then when it comes to muscle loss, a lot of people in general public and also many scientists, we use the word sarcopenia. Is there a specific definition of sarcopenia? I mean, having just less muscle mass is not necessarily.
Yeah. Well, I mean, it's a great point coming back to the concept of muscle quality, because for, a long time, the focus was on muscle mass. That was only what defined sarcopenia, was how much muscle that you had, the muscle mass. And I think studies like Health ABC were important because it really started to shift the paradigm to get people thinking more about muscle quality and function, not just muscle mass. Because, you could be a 70-year-old marathon runner, be, fairly thin, have relatively low muscle mass, but be quite healthy.
Yeah.
And your muscle quality is good. So it's pretty clear now that muscle mass is really not a good at least solely as a good
Yeah.
measure of health, right?
Yeah. So then after ABC, health ABC, is there anything else that you found in health ABC that is worth mentioning?
Well, so we did a very small study in Health ABC with muscle biopsies, and we looked at their mitochondria function. Some of these subjects had really, really good mitochondria function into their 90s. And so Health ABC was really instrumental in for our group to do the next study on muscle aging, which is called the study of muscle mobility and aging, or SOMA. And we're into our 5th year of that study, and we got muscle tissue samples from over 800 older subjects.
Wow.
And we're predicting in their muscle cells and their muscle tissue their trajectories of aging and mobility limitations. So that's an exciting study to be part of right now. Really taking Health ABC and the studies like that to the next level of really starting to interrogate the molecular biology and the biochemistry in skeletal muscle in aging, and following these older men and women over time to see how the muscle how the muscle cells are changing with age and how this relates to their function.
So when you said mitochondria function in this muscle, so did the mitochondria, how healthy are these mitochondria predicted muscle strength and quality?
That's what we're looking at in this current study.
Yeah.
And
But in the first study, ABC, did you see any?
yeah, we didn't have enough subjects. We just did a really small study. But this next study that we're doing now, called SOMA, I think we'll get some really good information on
How many people?
878 people.
What's the age range?
Yes.
Age is.
Age, they're over 65.
Yeah.
Yeah.
Okay, so they're a little bit younger than
Yeah.
health ABC.
Yeah.
And they have to do, what all they have to do in this study?
So in this study, they're doing, MRIs for body composition. They're having muscle and adipose tissue biopsies. They're having exercise tolerance tests, muscle function test, gait speed analysis, and many of the measures that you would want to do in older people to see what predicts, loss of function.
Yeah, so how much muscle biopsy do you take? Because when people hear about muscle biopsy or fat biopsy, they're thinking that somebody coming and grabbing a chunk of muscle.
Yeah, no, it's done. It's taken with the needle. It's a needle biopsy. It's about the size of a pencil eraser.
Okay. Yeah.
In terms of the amount of muscle tissue.
Yeah.
I've had many on myself, and
I know.
still walking around. So,
And then the fat is the belly fat.
the belly fat. Yeah, we take the adipose tissue from the belly. Yeah.
And then they get a full body MRI or.
Full body MRI.
So that's pretty good deal for people who are on this study.
Exactly. Have their blood work done and all that. So.
And then how regularly they have to come back?
Yeah, annual visits, but not every visit is an intensive visit, right? So the 3-year visit is more of an intensive visit with the repeat MRIs and exercise tolerance tests, and so forth. So
what is an exercise tolerance test?
Yeah, so an exercise tolerance test, we put people on a treadmill. You start out at your normal, comfortable walking speed, and then we simply increase the grade or the steepness of the treadmill until you can't exercise, until you can't walk anymore. And that's your maximal exercise tolerance. Yeah.
Are you also collecting VO2 max?
We are.
Yeah?
Yeah, it's a classic VO2 max test.
So what is the average VO2 max you are seeing in this SOMA study?
It's pretty low, Satchin. It's
20,
less than 20.
Less than 20?
Less than 20 milliliters per kg, yeah.
Okay, what would be your version of ideal, a healthy aging person?
For that age, I think 30 would be reasonable. 30 to
35?
35, yeah.
So 65 to 75 is, or 65 above.
And above, yeah.
So they can be 65 or 95, they can be part of this study?
Yes.
Oh, okay. Yeah. So then that gives you a lot of power to see how mitochondria function, muscle function, composition, everything.
Exactly. And I think this study will give us a window into what parts of the body might age better than others, right?
I see. Yeah.
I don't think everything in our body ages the same, right?
Yeah.
I exercise, but I don't have any hair, right? I mean, exercise can't do everything.
Exercise doesn't
Right?
grow hair.
Yeah.
You have done that experiment
Exactly.
long time.
Exactly.
Yeah.
Yeah.
So coming back to the mitochondria test,
Yeah.
like in the lab,
Yeah.
what do you actually test in mitochondria?
So we'll take part of the muscle sample and we'll put it in these instrument, and we provide the muscle tissue and the mitochondria the fuel for it to do what it does, which is consume oxygen.
Yeah.
Aerobic or oxidative metabolism, and we can measure this very accurately. How these mitochondria work, essentially.
So you essentially give this mitochondria sugar to eat and then
Sugar or fat,
Yeah.
and see how it oxidizes or consumes oxygen in response to the fuel.
Yeah. Is there any reference range to say that this is a healthy mitochondria, that's an unhealthy mitochondria?
Yeah, it's a good question. I mean, we certainly have values, right? We have younger reference values that we can look at. And, like a lot of other things, there's such a range in these older people. Some have poor mitochondria, some have better mitochondria.
Yeah.
And the interesting thing is going to be, how does that variability in mitochondria, how does that predict the trajectory of aging?
Yeah.
Right?
So you're looking at that mitochondria function also with the exercise tolerance test, because that is cardiopulmonary.
But certainly, it's certainly going to be related.
Right? And then that one, you're also connecting with the muscle strength.
Yeah, muscle strength.
Yeah, so that would be really cool, because a lot of people that talking about mitochondria, and there are a lot of supplements out there for mitochondria, but there is no definitive study so far that I can think of with large number of people like this, where we can connect how mitochondria function.
Exactly.
Yeah. So that's why these studies are extremely important. And
Yeah.
So you have already recruited 800 persons.
Yeah, and the plan moving forward with that study is that we're hopeful to get additional funding to keep the study going. And in addition to keeping the current participants in follow-up and measuring them over time, is that we're going to add over 400 new subjects to the cohort in Florida, where I am. And that'll help us enhance diversity of the cohort. We can start understanding the effects of aging in non-white Hispanics, et cetera. So,
and then there's one question that was coming to my mind, and now see, this is the effect of aging and jet lag.
Right.
No, what I was thinking was, you said you're trying to add 400 people from Florida.
Yeah.
So from where are you, have you recruited this 800 people who are already on?
So the 800 existing are from two sites, North Carolina and Pittsburgh, Pennsylvania.
Oh, so just like the Health ABC study.
Exactly.
I see. Okay. But they are not the same people, because we know that.
Not the same people. Well, Health ABC was Pittsburgh and Memphis, Tennessee.
Oh, Memphis, yes.
Yes.
Yeah, this one is. So 800 plus 400, that will be a large study. And then, so are there clinical sites there? They come there to.
Yes. Yeah.
So
clinical
sites.
Yes. Exactly. Three different sites.
Three different sites. And then now the most exciting thing that we're involved in, these are just longitudinal study, no intervention and nothing. And now MotorPack. We're also involved in MotorPack. So
MotorPack, the Molecular Transducers of Physical Activity Consortium, this nationwide exercise study that we're involved with is, we consider sort of the moonshot for exercise research, right? And we'll have people do either endurance exercise or resistance or weight training exercise to see how these different exercise programs affect muscle cells, fat cells, and really try to understand the variation of exercise. Like we were talking about with aging. We know that not everybody responds the same to exercise. So I think the power of this motor pack study will be to really understand on a molecular level, why is it that some people respond better to exercise than others.
Yeah.
So we're studying almost 2,000 subjects in this exercise study with muscle and fat biopsies and blood samples.
They're all healthy people.
All fairly healthy people. Yeah.
Yeah. So what are the inclusion, exclusion criteria?
Yeah, so it's across the adult age span, so there's no upper age limit. So we're studying, adults from the age 18 to, 80 plus.
Yeah.
There's also one site in California that's a pediatric motor pack site. Only blood samples there.
Yeah.
Yeah.
Yeah. And then these people will, some will do exercise and some will not do exercise. What is the idea?
Yeah, so it's a randomized controlled trial. Some are randomized to resistance exercise, some are randomized to endurance or aerobic exercise, and then some are randomized to a non-exercise group for control, a comparison, control comparison group. 12-week program. So we'll study them before and after the 12 weeks.
So they have to come to the site to exercise in front of the trainer?
It's all supervised. Yeah, so this study is really not to address how you get people to exercise.
No.
Right? This study is to address when you actually do the exercise, what happens.
Yeah. So they have to come there, show up for the exercise.
We're counting every heartbeat, every repetition of the weight stack. Yeah.
So when they come to the training center, so you put a heart rate monitor.
Heart rate monitor. They're wearing actigraphy, accelerometers. They're working with their personal trainer or exercise physiologist, sort of exercise coach. So they get their sort of own personal trainer for 12 weeks for this program.
Wow.
And to say that the subjects who do get randomized to the non-exercise group, we sort of view that as a wait list control because they get exercise after their 12 weeks.
And then how many times in a week?
3 times per week.
1 hour each?
About an hour, 45 minutes to an hour per session.
Yeah. It's not that bad.
No.
Oh. And then what are you measuring?
So we're measuring...
During exercise, what are you measuring?
During exercise, we're measuring what's going on in the muscle cells, what's going on in the fat cells. We're measuring, substances that are released in the blood. We're measuring body composition, muscle strength, fitness. Yes, we're measuring a lot of stuff.
How do you measure fitness?
It's the VO2 max test.
VO2 max.
Yeah, we're doing a VO2 max test, this time on a bike.
Yeah.
Yep.
And then have you looked at any of the age-specific VO2 max? Are you seeing the trend that
absolutely.
younger humans?
Yeah.
What is the trend?
Well, clearly older people have lower VO2 max, right?
Yeah.
Because I was showing some of the data here at this meeting, and the overall values look pretty low they're a sedentary population.
Yeah, but you're taking the average of 2,000 people, so...
And we have some older people that bring down the averages, right?
Yeah. So, but in the younger, you do see more than 35 and 40.
Yeah. Not often.
Not.
They're still pretty unfit.
Really?
Yeah.
And what is the BMI was 26 or 20?
26, 27 on average.
Yeah. That's not that bad, but it's still not the ideal BMI.
Yeah, exactly.
So how long will this study, how long will it take to get the results from this study?
Yeah, so we have about 1 year to go.
Yeah.
We're, I'd say, 2/3, probably more than 2/3 of the way through enrollment, and we have about a year to go.
Yeah.
And by the time we finish the actual human study, clinical studies, in a year from now, it'll probably take another 1.5 or 2 years to get all the analysis done.
Yeah. No, this will be exciting.
Yeah, very exciting.
There is no other. Is there any other comparable study on?
I don't. As a single study, I don't think so. I mean, people are starting to do some analyses, combining results from multiple studies.
Yeah.
Which is important as well, but I don't think there's a single study like MotorPack that's as big and comprehensive.
Yeah. And also the technology has changed now. So now even from the blood, you can actually see
Yeah.
how different organs are working because you'll have organ-specific
That's right.
signals in the plasma.
That's right.
Blood work.
Yeah, and MotorPack was set up to sort of be built for the future in terms of archiving extra samples to be analyzed later with as newer technologies emerge, right? So,
Yeah. And what do you do personally? I have heard that
yeah.
you are an Ironman for many times you have competed.
Yeah, I've done Ironman, I've done cycling races, marathons, continue to be active, sort of practice what I preach.
Yeah.
Again, that's partly why I got into this whole field because of my own personal interest in my own performance, right? My own aging.
Yeah.
I start to care more and more about. So, yeah.
So what do you do for to compete for Ironman? Like nutrition, sleep, exercise.
Try to eat, eat as much as I can to maintain my training when I'm training, 15 to 20 hours a week for Ironman.
Yeah.
And to be clear, I don't do that all year round.
No.
But for certain times, you train more than others. I think my sleep's better when I exercise that much. But I also feel like, my sleep is not as good as it used to be. When I was younger. So
that happens with everybody.
Yeah. And as you know, it's all tied in, right? Exercise, diet, sleep. It's hard to really disentangle those.
Yeah. I mean, there are studies showing that exercise actually improves sleep.
Yeah.
But at the same time, too much exercise may not help you well.
Yeah, exactly.
Yeah. So thank you so much, and I'm looking forward to the results from MotorPad.
No, thanks. It was a pleasure to talk to you.
Yeah. Thank you.
Good.